Hey, Mr. P here. In this video, we're going to talk about the principle of independent assortment and compare it to segregation, which was the purpose of the last video. If you recall, the principle of segregation talks about how alleles assort out from a gene. Remember, there is two alleles, or you should have two alleles per gene, and those alleles will separate out evenly. That is the principle of segregation, and so we're going to talk about now the principle of independent assortment, do another cross with pea plants, this time a dihybrid cross. You'll notice that our P generation in this video, or at least in this diagram, contains 4 total letters, 2 R's and 2 Y's. That means we have a dihybrid cross. We're looking at a cross with 2 traits, and we're going to talk about not only how those alleles separate out, but how the gametes are made, how the gametes come together, and ultimately how those genes and chromosomes separate out or assort independently of each other. Okay, this is the principle of independent assortment. Let's go. So Mendel's crosses involving 2 traits are called dihybrid. In the last video, we talked specifically about monohybrid, and I know in class we've talked about monohybrid and dihybrid. Monohybrid, remember, is a cross involving a single gene trait. Now this is 2 traits, which is a dihybrid. We can go trihybrid and even bigger than that. But we're going to focus for this, this time being in this video about dihybrid or on dihybrid crosses. That is a 2-trait cross. If we have 2 alleles per trait and 2 traits, we have a total of 4 alleles, 2 R's in this case and 2 Y's. Just like in our monohybrid studies or in Mendel's monohybrid studies, in the P generation, he used purebred or true breeding homozygous parents. And when I say homozygous, I mean it is completely the same allele in both cases, right? So in this particular parent, this is a true breeding homozygous dominant parent, 2 big R's, 2 big Y's. This is a true breeding homozygous recessive parent, 2 little R's and 2 little Y's. The way that we assort these traits or assort these traits independently to find our gametes or to produce gametes is we have to FOIL. F-O-I-L. Big R, big R, big Y, big Y. In this particular parent's case, FOIL, F-O-I-L. F stands for first, so it will be the first letter of each group of letters. So in this case, the big R and the big Y. That would be our first gamete. Outside would be the 2 outside, so that would be big R, big Y in this case. Inside for I, big R, big Y, and then last would be big R, big Y. So this particular individual will produce 100% big R, big Y gametes, which is why this is the only option shown. So this will be 100%. This one, if we quickly go through the independent assortment associated with these gametes or this gamete production, you'll notice that 100% of that individual's gametes will be little r, little y, because of the same phenomenon, the same fact, right? First, outer, inner, last. You get 100% little r, little y. Okay? Now, just like in his last or monohybrid crosses, we're going to take these parents, we're going to find these gametes, and then we're going to cross them to produce our F1 generation. Now, if we cross with a quick kind of branching, the F1 plants are going to produce, or the F1 plants that we cross are going to be 100% what we call heterozygous. Okay, which means they have a big and little. In this case, they're true heterozygotes, or they are heterozygous for the R gene and for the Y trait. Okay, now when we cross these individuals, 100% heterozygous cross, or in this case a true dihybrid cross, we're going to produce our F2 generation, which you can see here by this Punnett Square. Again, we have to find our gametes, and so in this case, if our heterozygous individual is big R, big Y, little y, we're going to FOIL exactly the same way we did up here to find our gametes, only in this case they are not going to be 100% identical. They will be, in this case, big R, big Y, as our first sperm cell, big R little y as our second, little r big Y as our third, and little y as our fourth. If we do the same thing with the egg cell, you'll notice that you get the exact same 4 options. And when you put them in a Punnett square, you will get 4 along the top and 4 along the side, giving us a 16-box Punnett square, which in this case is going to give us a 9331. Now we're going to talk a little bit more about why the 9331 is significant and why the 9331 is the expected ratio. But when Mendel conducted these 2-trait or dihybrid crosses, he utilized the pea plants, as we've already discussed, to produce a lot of offspring. So in this particular cross, he produced 556 peas with these particular phenotypes that are a combination of the 2 true breeding parents he set up to use in this cross from the very beginning. Okay, yellow being the dominant trait over green, and round being the dominant trait over wrinkled. Okay, so 9/16 will show both dominant traits, that is yellow and round. 3/16 will show a single dominant trait and the other recessive. In this case, the green is the recessive and round is the dominant. In this case, yellow is the dominant and wrinkled is the recessive. And then 1/16 is going to show both recessives, which in this case, green and wrinkled are both recessive. Okay? So, of the 556 total peas, if we identify or try to calculate how many we should get that show the 9331, or of the 9331 ratio of 556, 315 are smooth and yellow, 108 smooth and green, 101 wrinkled and yellow, 32 wrinkled and green. This is roughly a 9331, which gives us a total of 556 offspring. Okay? So, 315 plus 108 equals 423. Okay? Where do we get that number? That is the number that show the smooth phenotype. And 101 and 32 are the 2 that show wrinkled, and so that comes together to be 133 or 133. If we look at this specific ratio, 423 to 133 is really close to a 3-to-1. Okay, why is that significant? Well, smooth to wrinkled is the dominant to recessive for the R trait, okay, or the R gene. So big R was smooth, little r was wrinkled, and I can go little r because it had to be homozygous recessive in order to show wrinkled. If we talk about yellow, 315 plus 101 gives us 416. Where does the 315 and 101 come together? 315 and 101, those are the ones that are going to show yellow, which is the dominant trait. Green, which is the recessive trait, 108 and 32. Okay, green and green, 108 and 32. Those come together to give us roughly 140. If we put those into a ratio, 416 to 140 is very close to a 3-to-1. Again, that's really significant because yellow is the dominant, big Y, and little y. And so why are these individual genes giving us a 3-to-1 in both cases? Well, if you remember from our monohybrid crosses, that 3-to-1 is the expected ratio when you are just looking at a monohybrid cross. Remember, if I show the monohybrid cross in a single monohybrid Punnett square, and we cross, in this case, big R, little r, you will get big, 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 little, little, little. That is a 1, 2, 3 to 1 that will exhibit smooth to wrinkled. Okay, so 3/4 smooth, 1/4 wrinkled. Same thing over here. If I put the individual monohybrid cross together, big, little, you get big, 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 little, little. Again, 1, 2, 3 to 1. That gives you a 3-to-1 ratio. Using the probability, Mendel considered the inheritance of both traits simultaneously, just like we did, okay? And he said if we looked at just the smooth or the wrinkled, basically the P shape trait, we should get a 3-to-1 smooth to wrinkled. And if we look at yellow to green, which is the trait for P color, we should get a 3-to-1 ratio in that one as well, knowing or utilizing the probabilities and the mode of inheritance that he already established in his monohybrid crosses. Okay, 3/4 smooth times 3/4 yellow, which is basically this condition times this condition, should give us 3 times 3 is 9, 4 times 4 is 16, 9/16 of the P should be smooth and yellow. Okay, because we know that the law of probability, and using the law of probability, tells us that an event that is combined is just the probability or the product of the two individual events. Okay, 3/4 smooth times 1/4 green should give us 3/16. 3 times 1 is 3, 4 times 4 is 16. That is the probability of smooth and green. Again, smooth is the dominant trait, so that's 3/4. Green is the recessive, so that is 1/4. 1/4 wrinkled times 3/4 yellow. That is the dominant trait. This is the recessive trait. Should be 3/16. 1 times 3 is 3. 4 times 4 is 16. That is the probability, 3/16, of producing a wrinkled and yellow P. And then 1/4 times 1/4 gives us 1/16. 1 times 1 is 1. 4 times 4 is 16. That is the probability of producing a wrinkled and green P, which again is both recessive conditions, which is 1/16. So when you put those together, the two individual monohybrid events combine to produce a dihybrid event, which is just the probability or the product of the two individual events. You get 9, 3, 3, 1, which can actually be seen pictorially in a dihybrid Punnett square. You will see that 1, 2, 3, 4, 5, 6, 7, 8, 9 will be yellow and round. 3 are yellow and wrinkled. 3 are green and round. And 1 is green and wrinkled. Okay? It doesn't matter if we use the law of probability and math to solve, or if we use a Punnett square and show pictures to solve. We get a 9, 3, 3, 1 every time in a true heterozygous dihybrid cross. Okay? So, how do we solve this type of problem using the fork line or branching method? This is one of the methods that you absolutely need to be familiar with and know how to use. So, let's look at solving a problem. The first thing you need to do is you need to identify your parents. Okay? In this particular case, our P generation is going to be homozygous dominant parent and homozygous recessive parent. When those come together, utilizing these gametes, that is 100% of her gametes, this is 100% of his gametes, that produces 100% heterozygous, or big R, big Y, little y offspring. If we self-cross that F1 individual, we get this cross over here. It's a true dihybrid, and it's a true heterozygous cross. Okay? First thing that you need to do is you need to identify the gametes. We FOIL to find the gametes, and the gametes are as follows. Okay? Big R, big Y, big R, little y, little r, big Y, little r, little y. Now, I'm going to go ahead and show you a shortcut because I think this is one of the easiest ways to solve these problems. You can either FOIL and find the gametes, and then you can cross the gametes with the gametes, okay? The female gametes with the male gametes. I like to go ahead and just separate the R's and the Y's, okay? We know that if we separate the R's, this is our cross, okay? What is the outcome of this cross? If you threw that into a Punnett square, you would know that big R, little r gives you big, 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 little, little, little. What is the probability? It's 1/4 big R, 1/2 or 2/4 big R, little r, and 1/4 little r, little r. these are the 3 genotypic outcomes of this cross, which is half of our cross, okay? What are we going to branch it with? We're going to branch it with our Y outcomes. So we know that our Y cross is that. If we throw that into this Punnett square, big Y, big Y, big little, 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 we get the exact same outcome. So 1/4 should be big, big. 1/2 should be big, little. And 1/4 should be little y, little y. What we're going to do in this Forkliner branching method is we're going to take these 3 y options and we're going to branch them off of every r option. Okay, so we're going to take these same 4 options, 1/4 big Y, and we're going to put them down here as well. Okay, so now we have our 3 Y genotypes branched off of each of our 3 R genotypes. And now we can just combine them. Now we're saying that if this outcome mixes with this outcome, it gives us this outcome. 1/4 times 1/4 equals 1/16, big R, big R, big Y, big Y. Okay, put this one together. 1/4 times 1/2 is 2/16. Big R, big R, big Y, little y. Okay, big, 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 little. One quarter times 1/4, 1/16. Big R, little y, little y. Okay, and we just continue to put them together. 1/2 times 1/4 is 2/16. Big R, big Y, big Y. 1/2 times 1/2, 4/16, or 1/4. Big, little, big, little. 1/2 times 1/4, 2/16, big R, little y, little y. 1/16, little r, big Y, big Y. 2/16, little r, big Y, little y. And the last one, 1/16, little r, little y, little y. Okay? These are the 3 R outcomes. These are the 3 Y outcomes. These are the total genotypes of this particular dihybrid cross. Okay? Now, what do you have to do? You have to link all similar phenotypes. Notice that these are all different genotypes, but what they all have in common is they both are going to express the trait that the big R and big Y code for. In this case, they're going to be round and green, okay, or round and yellow, okay, round and yellow, just like this original parent. So we put those together and we add them up. In this case, 1 plus 2 is 3, plus 2 is 5, plus 4 is 9. 9/16 will be yellow and round. Okay, then we combine these two, which are big R, little y. 1 plus 2 is 3, so that's going to make 3/16 are going to be green and round. Then we combine these two, which are little r's and big y's. And so phenotypically, that's going to be 3/16 yellow and wrinkled. And then this one is the last one. It is homozygous recessive. It is the only one that is homozygous recessive. It is little r and little y, so it'll be 1/16 and it will be green and wrinkled. Okay? Again, 9:331 is our expected ratio. That is the ratio of our F2 phenotype. Okay? There is a difference between F2 genotypic ratio and the F2 phenotypic ratio. The phenotypic ratio is the ratio of what they physically look like. The genotypic ratio is the actual genetic makeup or the genetic constitution of the particular organisms or offspring. And so if we focus on putting these into an actual ratio, the F2 genotypic ratio of a true dihybrid heterozygous cross will actually be 1, 2, 1, 2, 4, 2, 1, 2, 1 every time. 1, 2, 1, 2, 4, 2, 1, 2, 1, which will give us the phenotypic ratio of 9:331. Okay? Chi-square analysis is used to then evaluate how closely the results of the cross fit our expectations. We're obviously going to do some chi-square analysis and some sample chi-square problems in class. Bring those questions to class. See ya.